All right, here in chapter 21, we are going to wrap up the cardiovascular system by talking about the blood vessels and circulation. So first of all, the functions of the circulatory system. So we've already covered this in chapter 20, but just to hit on it again, of course, the pulmonary vessels, those are transporting blood to the lungs. They're going to be picking up oxygen. Then the systemic vessels, those are going to be transporting blood from the left ventricle to all parts of that body into the right atrium. So the functions of the circulatory, of course, carrying blood to the tissues and back, exchanging nutrients and waste products, transporting lots of different substances like hormones, enzymes, nutrients, gases. Also, more what we'll focus on here is regulating blood pressure, and then also directing the blood flow to tissues by controlling the blood volume. So looking at the blood vessels. So there are three different types of vessels that form this passageway from the heart to the tissues and then back. So of course we start with the heart. Doesn't count as a vessel. But then the heart, everything going away from the heart are called those arteries. So that's one type of vessels. Then we have thin layers that are capillaries, and then from the capillaries, the blood is picked up and taken back to the heart with the veins. So the blood vessels are arteries, capillaries, and veins, and then back to the heart. So if we look at the structure of the vessels, looking at the different tissue layers, the first layer, the tunica intima, so we think about internal or intima. So this is the endothelial, so that tells us as well as inside. That's going to provide that basement membrane, and we have that connective tissue layer called the lamina propria. And then we have that internal elastic membrane, and what are called fenestrated layers of elastic fibers. Then we have the tunica media. So this is going to be the smooth muscle cells that are arranged circularly around the blood vessels. This also has some amounts of elastic and collagen fibers, depending on the size of the vessel. And then a couple terms that we'll use, which we've already used a little bit. Vasoconstriction. This is when those smooth muscles contract. That's going to decrease blood flow. So think about squeezing on a pipe, making it smaller, which is going to decrease the flow through that. Then we have vasodilation, which is the opposite. This is where those muscles relax, and that will increase the blood flow by allowing a larger diameter and more area for the blood to flow. And then the final layer is the tunica externa. So we have the interna and externa. This is, again, connective tissue. It varies anywhere from dense, regular, near the vessel to kind of a loose connective tissue that kind of surrounding that connective tissue. So if we look at these three layers, again, on the outside, we have that tunica externa there with that connective tissue, some of that loose connective tissue. Then inside of that, the tunica media, where we have this elastic membrane and smooth muscle that allows that vasoconstriction during contraction or dilation during relaxation. And then we have the tunica intima all the way inside, which has the endothelium. Then we have this basement membrane with the lamina propria, and then that elastic membrane that then connects it to the tunica media. So if we compare our arteries and veins here, a couple things that you can see right off the bat. So our arteries here indicated as the A. So the tunica intima looks pretty similar. It's a pretty thin layer. Where we start to see differences, though, is how much thicker the tunica media and tunica externa are, especially the tunica externa in the arteries. They're going to be much thicker. So we're going to have more smooth muscle, more connective tissue that allows that constriction and relaxation to control blood flow. So if we look at these starting with our arteries, the different types of arteries. Again, arteries by definition are carrying blood away from the heart. So they're going to get smaller as they go further and further away. We have a couple different classes of arteries: elastic arteries, muscular arteries, and arterioles. So as the arteries get smaller, they transition from a large amount of elastic fibers, so they're bigger, more elastic, to smaller amounts of smooth muscle, to the opposite. So as they get smaller, we have less elastic fibers and more smooth muscle. So the larger the diameter, the more elastic fibers and the less smooth muscle we have. So if we look at those elastic arteries, these are also called conducting arteries. These are going to have the largest diameter. Again, bigger means more elastic. So that means that we have high pressure, so that pressure is high and fluctuates between the systolic and diastolic pressures. So we go back and forth. Again, more elastic tissue than in those muscles. Now, than the smooth muscle, sorry. So relatively thick tunica intima. So again, that internal part, and then a thin externa. So thicker internal part layer of those arteries, thinner on the outside. And typically the name includes most of the named arteries. So most of the ones you'll be learning about in lab, these are going to be more elastic arteries. Then the muscular arteries, they're named this because the smooth muscle allows the vessels to regulate the blood supply by constricting and dilating. So most of the smaller unnamed arteries are going to be more muscular arteries. So we won't focus too much on those in lab. We won't be naming a lot of those. But again, they have a thick wall due to all that extra smooth muscle, hence the name muscular arteries. They have lots of smooth muscle. But again, they're smaller in diameter. So these are also called distributing arteries because that smooth muscle allows the vessel to partially regulate the blood supply. And again, these are going to be smaller arteries that are adapted for vasodilation and vasoconstriction. And then our last type of artery here, the arterioles, these transport blood from small arteries to capillaries. So we go kind of more our elastic to the muscular to the arterioles and then into those capillaries. If we kind of think about diameter. So these are the smallest arteries, and this is where the three tunica can be actually differentiated. Past that, we move into capillaries where we can't tell the different layers. So like the small arteries, though, they can control that vasoconstriction and dilation. Then we move into our capillaries, and this consists of endothelium, so that simple squamous. They do have that basement membrane, and then a small kind of delicate layer of loose connective tissue, but we're not going to have those three layers like we saw with our arteries. So they have scattered what are called pericapillary cells that are fibroblasts. They have macrophages or some undifferentiated smooth muscle cells. And these vessels, their job is for exchange between the blood and the interstitial space. So for those gases to be able to pass through, it needs to be a thinner layer. They can't move through all three of those layers we were just talking about with the arteries, so it's harder for any exchange to happen. So that's mainly going to happen in these capillaries. A few different types of capillaries. So we have continuous capillaries because there are no gaps between the endothelial cell. There are no what are called fenestrae. So they're less permeable to large molecules than our other capillaries. So like muscular and nervous tissue, not a lot of larger molecules can move through them because they don't have those gaps. It's a kind of a tighter connection. Then the fenestrated, these endothelia have these fenestra, which are areas where the cytoplasm are absent and the plasma membrane is made up of thin porous diaphragm there. So it is going to be very highly permeable. So think about like the villi that are in our lungs or in the intestines. and then the glomerula of the kidney, where we have a lot of exchange going on. Again, very, very thin membrane, allows for lots of exchange, highly permeable. And then we have the sinusoidal, which have a large diameter. They also have those sphincterae, which are much larger, less of a basement membrane, and this is mainly going to be our endocrine glands, seen also in the liver as well. So substances cross those capillary walls by diffusion, either through or between the cells, or more commonly in those sphinctera. That's why the continuous doesn't have as much permeability, because they lack that service. Right, and this is just a nice picture here that shows that, again, the continuous, we don't have any of these sphinctera with the diaphragm there, and then we have over here one without the diaphragm, just again a complete opening here versus a small little diaphragm that kind of holds them together. Both are considered fenestrated capillaries, but again, we can have much more gas exchange, exchange of nutrients here that we don't get with this continuous capillary that's lacking those. And then the sinusoidal have even larger openings, or those sphinctera. All right, so here in our capillary network, we're going to have the blood flowing down through that arteriole. It's going to branch off into what's called a meta-arteriole. It's going to flow through what's called the thoroughfare channel, and then into the capillary branches that we see here, and into those venous capillaries, and then out into the venule, which is going to return the blood to the heart. So then all of the gas exchange is going to be happening here in this branching network between the arterial capillaries and the venous capillaries. That's where the thin walls can occur that allow that exchange of nutrients and gases. There's also what's called the precapillary sphincter, so you can see, let me change my color actually, so that you can see them. But some of these sphincters, there's one here, here's another one that's being shown. But these precapillary sphincters, what they are going to do is they're going to help to control or regulate the blood flow into that network. Because what's going to happen is that in the thoroughfare channel, that's to the venule, that's a fairly consistent flow. However, in the arterial capillaries, it's a little bit more intermittent flow through there. All right, so if we look at differences in tissues that have a lot of metabolisms, anything about like our muscles, they're going to have more capillary networks. So again, our lungs, the liver, the kidneys, the muscles, because they need a lot of oxygen, a lot of nutrients, they're going to have a lot more of those capillary networks than what other regions may have. So those networks in the skin, they'll help with regulation. So a little bit less about nutrient and oxygen demand, but it helps to provide a loss of heat whenever we're sweating, and to kind of regulate our temperature. And then the major function, though, is that nutrient and waste exchange. So dropping off glucose and fats to allow for metabolism, and picking up waste as well. All right, now if we switch over to the different types of veins, so just like we have 3 types of arteries, we have 3 different veins. We have venules, small veins, and then medium or large veins. And again, these are carrying blood toward the heart. Now, typically this is deoxygenated blood, and arteries have oxygenated. Again, the exception to that, though, are the pulmonary arteries and veins, where the pulmonary veins are carrying blood to the heart, but it's oxygenated. Pulmonary arteries carrying it away from the heart, but it's deoxygenated, waiting to pick up that oxygen. So if we look at the venules, these are what we just looked at with the capillary networks. They drain into that, so that those endothelial cells, they have that basement membrane and just a little bit of smooth muscle. So as the diameter, though, increases, just like with our arteries, the smooth muscle is also going to increase as well. But these are pretty small, pretty thin comparatively, that feed out of those capillary networks. Then our small veins have that smooth muscle, form a continuous layer. It's not as intermittent as with our venules. So now we have the addition of a layer called the tunica adventia that's made up of collagenous connective tissue. So we have that tunica intima that we had also with the venules, but now we've added tunica adventia to it as well. Then those medium veins are kind of the go-between between that small and large vein. And then finally our large veins, that tunica intima is very thin, so a thin endothelial layer, relatively thin connective tissue, but now we're going to add in those those elastic fibers. So the tunica media is circularly arranged, and then the externa is the predominant layer, the thickest of the layers in those large veins. Okay, we also have what are called portal veins, and portal veins begin kind of in a primary capillary network that extends some distance and then at a second capillary network. So they are essentially going from one capillary network to another that's over a pretty long distance without a pumping mechanism, meaning there's not like a heart or anything like that in between it. We have 3 portal veins in the body. The main one that we talk about is the hepatic portal vein, and this is going to carry nutrient-rich blood from the GI system, so from our intestines, to the liver. So again, it goes straight from the gut to the liver, not the heart in between, so the capillaries of the gut to the capillaries of the liver, where they dilate into those sinusoid capillaries with those large fenestra that allow for lots of exchange. So it's taking a lot of nutrients straight from the gut right to the liver. Excuse me. The 2 other ones, so we have the hypophyseal, rolls off the tongue, portal system, and that goes between the hypothalamus and the anterior pituitary gland. So talking more about that in the endocrine chapter as well. But that's going to be a portal system that allows for hormonal control between that hypothalamus and the anterior pituitary, again going from capillary bed of one to the other. And then final, the renal nephron portal. This is within the urine, producing structures of the kidneys, where again we're getting exchange from one capillary network to the next. Then we also have some valves here. So of course we talked about the valves in the heart. We also have some valves that are found in the veins that are greater than a couple millimeters in diameter. Not that you would need to know that number, but just to put it in perspective, folds in the intima are going to form the 2 flaps, essentially that overlap. So again, that intima layer, the layer all the way on the inside, essentially that layer is going to fold over, and as it folds over, it creates a valve structure. So more valves are in the veins in the lower extremities than in the upper extremities to help ensure proper blood flow. Right, and then kind of wrapping this part up here, so the neuronal innervation, so think about the neurons coming into these vessels. So we have unmyelinated, so again, myelin helps that conduction occur. So unmyelinated, sympathetic, again, fight or flight, nerve fibers form a plexa of tunica adventia that allows for vasoconstriction. So then we have small arteries and arterioles have the greatest extent of innervation, receiving the most nerves coming in. Vessels of the penis and the clitoris are innervated by parasympathetic rather than sympathetic neurons. And then some of the vessels are innervated by myelinated fibers, and they act as what are called baroreceptors, looking more at pressure changes, so detecting that blood pressure and sending that information to the brain.